principal component analysis. Consider this motivating example. Some patients walk into your clinic and they measure their mass and their height. So if you plot the square of the height on one axis y here and as well the mass, you will note that there's a strong correlating trend between those variables. So our idea here is to try to capture as much of this variance inside of one single variable. That is, that we want to find the direction that describes most of the spread in the data. So we call that the principal component number 1. What that means is that you will note that there's a strong correlating trend between those two variables according to that axis, and that will describe most of the variation of their data. Now we can sort of look at, we can project every data point here back to that principal component, and this would now allow us to register the weight that every single patient, or the amount of variation that each patient has in this direction. Furthermore, we can look on the residues from any such projection to the principal component number 1. These will form sort of they will always be orthogonal now to the principal component number 1, and these often referred to as principal component number 2, or rather, if we take in this high dimensional space that's left, in this case it's just one dimension left, we would now find the axis of variation of the remaining of the residues when we calculated away the principal component number 1. You see that this trend is now going up with height and down with mass, and as a matter of fact, this is just the inverse of in this case, it just happens to be the inverse of what you probably have heard of before, namely the BMI of the patients. So the reason why we look at BMIs are normally that we sort of care more about the spread that does not sort of involve the component of the direct proportionality to the height of the patients. So the principal component number 2 here seems to be more interesting. That is the direction where we can see the spread now in BMI. A high BMI is normally seen as a bad thing, and this would come down now here in the negative direction here, according to principal component number 2. Consider a scenario where we have measured 3 different variables for patients. So it's variable X, Y, and Z. And the scenario is sort of drawn the way that there's a strong correlating tendency here between variables, both variable Z and Y, and variable X and Z, and variable X and X and Y. So all 3 variables are strongly correlating. Whenever there's a high value for variable X, there's a high value for value Y and variable Z as well. This is an interesting case for principal component analysis, because we would expect now to capture the main source of variation, which would be the source of variation that's sort of causing an increase in all 3 variables at the same time. So let's start with plotting the first principal component. Okay, so here's the same data now, but with the first principal component aligned. So the principal component now goes in the same direction as this data is covariating. Okay, so the next components will sort of cover the rest of the variation. So that might be now the directions in the 2 opposite directions. And remember, these principal components are always orthogonal to each other. So here it's the same data points again, but now with all 3 principal components aligned here. So we'll see now while the first component in blue sticks in one direction, the other ones are coming out of orthogonal to this. In this case, there's very little difference between the the directionality for the residues once you compensate for the first component. So the second component here in red seems to be as important as the green almost. However, the endeavor here is often that we want to compress this into a lower dimensionality. So in this case, we will just plot the first and second component in a separate plot. So here we plot the same principal component or the same data again, but in a two-dimensional plot. In this case, we see the first eigen component or the first eigenvariable here plotted along the x-axis, and the second eigenvariable on the y-axis. So it's no surprise here to see that the data is varying more in the according to x-axis, the eigenvariable 1, than the eigenvariable 2. And remember, that's what we saw in the original data as well. If we plot the eigenvariable number 3 here, it will look pretty similar to this one. It's worth noting that the dot product between the two principal components is 0 as well, or very close to 0. And that's just a consequence of them being orthogonal. There's this trick we can use from linear algebra. This is called singular value decomposition, SVD for short. In SVD, you can express a matrix as a product between two different or three different matrices, actually, where the middle one is just a diagonal matrix. Matrix X into a U and S and V matrix. We can see that the columns in the U matrix, they will form the projections of the first principal component of our samples in X, and likewise the rows in the V matrix is giving the projections onto these different samples inside of X. And this as well scales now with a principal component or the magnitude vector that's given in S. For the case where we have genes here as rows, and we have different samples as columns. This will now, if we would do a SVD on this matrix X, we would then get firstly the projections of the different genes. What would it looks like? What would a sample that describes the most of the variation in this matrix? It will sort of go according to the first column in the U matrix, and we'll do so with the magnitude that's given by the first value here in the single value matrix, the S. And it would do as well if we project the same thing in the same direction now, and would look now instead what would the typical gene look like that's described the most of the variation. That would still be described by the first principal component. That's given by the first row now in our matrix V. So this, by looking at this kind of data, we can start, or by doing this kind of projections, we can start to describe the variation in a data set. I will show you a short example. In the Jupyter notebook that we made for this exercise, I have put in PCA analysis of two different lung cancer data sets. Both these are downloaded from TCGA, and one of them is lung adenocarcinoma, and the other is a lung squamous cell carcinoma. So these are two types of cancer. If we combine this into one single data set, and we can calculate the first principal components, and if we would now sort of plot these first principal components against each other, we'll note now here for the eigengenes, that is the columns in the U. Here do we have firstly the first column in U and the second column in U. you will see if we plot now our samples, we note that the two different cancer types, the patients from the two different cancer types, they seem to be well separated, or not well separated, but there seems to be a trend where the single cell or the yellows, the patients here, they tend to be more high, or they tend to be higher on the eigengene number 1, and there's as well a weak trend here where the yellow patients here are going downwards as well on eigengene 2. The analysis allows us as well to do a second type of analysis. We can plot the eigenpatients as well of this data. So this again is the same data, but seen out of the perspective now of what would the patient look like, that is would describe the variation per patient that would describe most of the variation. And here we see the different genes now plotted according to the eigenpatient number 1 and eigenpatient number 2. And as a matter of fact, we can sort of now see that there's a couple of genes here that seem to describe very much of variation. So we could sort of investigate now the maximal genes and minimal genes according to this. So if we first look on the maximal genes, we see that there's two for eigenpatient number 1, the direction of eigenpatient number 1, we see one gene SFTPB and another, according to other component, we see KRT17 being on top. That's a carotene. The same carotene as well comes up in the as the most extreme component when we look on the minimal according to eigenpatient number 1. So the minimal eigenpatient number 1 is as well the maximal eigenpatient number 2. So that must be this data point up here. And the maximal eigenpatient number 1 seems to be this SFTPB, which probably is found over here. And we probably have a minimal eigenpatient number 2 somewhere among these genes. So these would be the genes that describe most of the difference between the data. Please remember here that this analysis has been completely unsupervised. So we haven't had any, we never instructed our algorithms to try to separate this according to different patient groups or anything like that. And that's great actually, because it doesn't sort of, this means that this algorithm is not prone to overfit in any way. So we can actually use this as an input for further analysis. We can, for instance, feed these two different these different directions and these different projections into further classification or statistical testing, etc. So it's a very powerful type of analysis because it really doesn't cost anything in terms of telling the algorithm what the actual ground truth is in a way. And you can as well see cases where you see a perfect separation between different patient classes as well. That means that you have set up you have managed to measure a difference in the experiment that seemed to be telltale by the different patient groups as well. And that's of course what you actually want to see. This is a great situation. I must emphasize though that in this case it's not for sure that this is actually what's happening. It might as well be that there's different biases in the different datasets. For instance, these datasets are probably processed separately, so they've probably been normalized separately, and that's a more likely cause of the difference here, and then the actual underlying difference in the different cancer types.